So now I'm going to show how to conduct this DNA protein cross-link reaction by the reductamination. So for this I'm using histone S3 protein as unmodified protein, and I'm using 5-4-mil-DC containing DNA as its counterpart. So to conduct the DNA protein cross-link, in contrast to peptide, the ratio of DNA to protein are 1:2. So if you take 500 picomole of DNA, then you use like 1 nanomole of protein, so that the reaction proceeds smoothly. The reaction conditions are almost same in DNA peptide conjugation. So where I use this sodium phosphate buffer with the 5 millimolar, 10 millimolar sodium phosphate buffer, 10 microliter. And then to that I add 500 picomole of DNA, which is like 2 microliters. And then I use 4 microliter of histone S3 protein, so which is stored here, and then 4 microliter of water, so the total volume of this reaction mixture is 20 microliters. So after adding everything, and then you mix it properly, and then put the reaction at 37 degree for 3 hours. So then we can take that now again if you want to start the next step. After 3 hours of the reaction, take this reaction mixture out and then add 5 microliter of sodium cyanoborohydride to the reaction mixture, and then mix it properly. After mixing, and then keep the reaction running at 37 degree overnight, or which is close to 16 hours. After the conjugation reaction for overnight at 37 degrees Celsius, the DNA protein conjugate has been dried on the speed vac. After that, this reaction mixture was reconstituted in the water. So then there are two ways DNA protein crosslinks can be purified, either by the HPLC or by the SDS page. So purifying this DNA protein crosslink by SDS page is much easier compared to the HPLC. So for that, what I do is I take 4 to 12% SDS page. I mix the DNA protein crosslink with a loading dye in 1:1, and then heat at 90 degree for 10 to 15 minutes. After heating the reaction mixture, and then spin it, and then slowly add onto the SDS page, and then run the gels in like 1x TBE for an hour or so. So where you see the clear separation between the unreacted protein as well as the DNA protein crosslink. So then after running the gel separation, I wash the gel like 3 to 4 times with the water, and then I add the cyber green to stain the DNA, and then after 1 hour staining, I wash the stain for a couple of hours so that all the cyber green staining goes away, and then I add the simply blue stain to stain the protein part of the DPC. So after overnight staining with the simply blue, so next day I wash out the rest of this simply blue stain, and then I visualize this DNA protein crosslinks. So by Typhoon, I can just directly take a photo by the phone, or I can use the Leica to scan these gels. So once, depending on the mobility of these bands, like once I know which band corresponds to the DPC, when I correlate that with the protein ladder, so then cut that DNA protein crosslink band, and then put them into, transfer that pieces, the gel pieces, into clean Eppendorf tube. Then macerate these gel pieces within a plastic vessel. So technically what you do is take this gel piece containing this Eppendorf tube, put it into the ice bath, and then take this plastic vessels. So this is a plastic vessel, and then you slowly macerate the gel pieces, slowly putting here into this. So once you macerate it completely, add ammonium bicarbonate buffer with containing 0.1% SDS. So then take this out, which is clean, and then you vortex on the vortexer for 10 minutes. So then by doing that, what you do is all this DNA protein crosslink comes into the ammonium bicarbonate buffer. So then take this Eppendorf tube and then spin down at 14,000 G for 5 minutes. so that all the gel pieces settle at the bottom, and then you can carefully take the solvent from this tube which contains your DNA protein crosslink. After that, you concentrate this DNA protein crosslink, reconstitute it in water, and then desalt this DNA protein crosslink by the Amicon 3K filter. So then the Amicon 3K filter will give a very good recovery. 70 to 80% of the compound you can recover from the Amicon 3K filter. So if you want to use this for the mass spec, so maybe I prefer to do this Amicon 3K filter 2 to 3 times, so that none of this SDS or other salts will go onto the mass spec. And then you can characterize this DNA protein crosslink in a positive mode, since protein is a bigger part here compared to the DNA, and the positive charges are more, so that you run the DNA protein crosslink on a positive mode on the mass spec.